This program is brought to you by Stanford University. Please visit us at stanford.edu.
Well, thank you for that very nice introduction. You are carrying some of my workload, I think, for me, so thank you. I'm very glad to be here. I've been at Stanford now for about a year, and it really is good to be back. And what I'd like to do today is to, as Johanna told you, speak to you about hedgehog signaling, and I'll say something about its role in developmental patterning, but I'd like to emphasize in particular its role in neoplastic growth. And so, as already indicated, let's see, good, and okay, very good. The hedgehog gene was first described, its embryonic phenotype, in Drosophila, where the consequence of mutating hedgehog is that instead of the normal pattern of bristles on the ventral surface of the larva, with bristles alternating with patches of naked cuticle in each segment, the naked cuticle is lost in the mutant, giving rise to a continuous lawn of denticles which look to Nusslein-Volhard and Wieschaus a bit like a hedgehog. Well, this is a periodic defect, that is, every segment lacks a particular set of cells, and consistent with that periodic defect, hedgehog, which is stained for here in green, is expressed in a particular stripe of cells within each segment. This stripe is immediately adjacent to the wingless cells. And so hedgehog, as well as the Wnt signaling pathway, which you'll hear much more about on Friday, specifically from Roel Nusse, these pathways play roles in pattern formation in embryos. They provide positional signals that specify cell fate. They also regulate proliferation in a variety of tissues, and loss of signaling activity produces birth defects. In more recent years, attention has shifted from these embryonic roles of these pathways to post-embryonic roles in tissue homeostasis, in injury repair and regeneration, and I think it would be fair to refer to these kinds of activities as pattern maintenance as opposed to pattern formation, which is what happens in embryogenesis. And many of these activities occur through modulation of stem or progenitor cell physiology. And a lot of this work is just emerging. It really, though, is, I would say, the major focus of activity now in both of these pathways, that is, post-embryonic activity. And of course, as Johanna has mentioned, pathological activation occurs in pediatric cancers, endodermal cancers even, and this is just emerging now, a number of hematopoietic malignancies. So to now provide you with a little bit of background to the hedgehog pathway in the vertebrate, what you see here is a chick embryo stained for expression of the sonic hedgehog protein. Sonic hedgehog is one of 3 major hedgehog proteins in amniotes, birds and mammals. And you can see that it's prominently expressed in the midline, the notochord and floor plate of this embryo. Shown here are a series of chick embryos, a normal embryo on the left, and you can note here the normal optic vesicles. These bulges are the facial processes. This large dome is the forebrain in the developing chick embryo. And then this series of embryos here, where you can see that midline structures are progressively lost in these increasingly severe phenotypes. And this is due to the treatment of these embryos with a compound which has been mentioned already, cyclopamine, and this is a specific hedgehog pathway antagonist, and I'll say more about that a little bit later. The consequence of loss of hedgehog signaling, midline structures are lost so that these optic vesicles develop closer and closer together, and here a single optic vesicle, a fused optic vesicle, develops in the midline, displaces the nasal rudiments upward so that instead of 2 nostrils or olfactory pits at this stage of development. You now have a single nostril at the tip of a proboscis-like structure. And this is cyclopia. And actually, this is very, very similar to the phenotype of the sonic hedgehog knockout mouse, which we made and analyzed now more than 10 years ago. These kinds of malformations also occur in people. Here you see perfect cyclopia. And then here you see a series of less severe forms of what's termed holoprosencephaly. This is a term that gathers a whole range of malformations from the very severe cyclopia you see here to milder midline malformations. Here, for example, the eyes are still close. They're separate, but they're close set, and there's a single nostril. Here there's bilateral cleft lip and palate. This gentleman here has close set eyes, and the only other apparent defect is that there's a single upper midline incisor. So this is a less severe manifestation of holoprosencephaly. And the term holoprosencephaly actually derives from the internal anatomy of the severe form of this disease or this syndrome. And it refers to the fact that the prosencephalon or forebrain develops as an undivided vesicle, and here you see such a thing. So instead of the telencephalon, which will form the normal lobes of the cortex, and the diencephalon, there's one undivided forebrain structure or holosphere, hence the term holoprosencephaly. Well, in addition to these roles in differentiation and patterning of embryonic structures, hedgehog can also, in some settings, stimulate proliferation. And I'd like to use here to illustrate this point some work that was carried out here at Stanford in Matt Scott's lab. And I'll make actually a number of references to work carried out in Matt Scott's lab. He's made tremendous contributions to our understanding of this pathway and in particular to its role in cancer. So hedgehog signaling can stimulate cell proliferation in the developing cerebellum. And schematically here, hedgehog is expressed in the Purkinje cells, and it acts to stimulate proliferation of the external granule layer, the precursors of cerebellar granule cells. And here, just to illustrate that, what you see is that in a cerebellar slice, a control slice, there's very little BRDU incorporated. However, if that's incubated with sonic hedgehog protein, there is a very dramatic incorporation of BRDU. If you actually take these cells out of the cerebellum and plate them, put them in culture, they become highly dependent on hedgehog stimulation for incorporation of tritiated thymidine, and that's what you see here. And so here's an example of proliferation that is regulated, stimulated by hedgehog signaling. And now you've already seen a form of this, but let me just repeat it to reinforce it and to be able to draw on it as we move along. In the absence of hedgehog signal, the multipass transmembrane protein patched, it's really very much like a transporter, and it's a member of a transporter family, inhibits smoothened activity. Smoothened is a 7-transmembrane protein, a member of the larger G protein coupled receptor family, although its embryonic patterning activities are not mediated for the most part by G proteins. When hedgehog is present, it binds to patched with the aid of a co-receptor and perhaps other co-receptors as well. This is a family of newly discovered co-receptors, IHOG in flies, CDO or brother of CDO in mammals. And this interaction of hedgehog with patched blocks the inhibition of smoothened, so that smoothened is then free to stimulate or to activate the pathway, acting through a family of cytoplasmic transcription factors, the GLE proteins, which upon activation then move to the nucleus and activate various targets, including actually patched itself. So patch is a target for the pathway. It's also a negative player in the pathway, and this is a feedback loop that serves to help keep the pathway regulated. Now, Gorlin syndrome or basal cell nevus syndrome was already mentioned. And this is a condition in which a patient is heterozygous for a mutation at the patched gene that inactivates patched, and is therefore subject to full inactivation of patched through a single additional hit. And when this happens, smoothened is no longer regulated, becomes constitutively active, and activates the pathway, and this results in formation of various tumors. Basal cell carcinoma, for one, and here you see a Gorlin syndrome patient with a plethora of lesions, skin lesions. These are basal cell carcinomas, and all Gorlin syndrome patients do get basal cell carcinomas, particularly on sun-exposed regions of their skin. A small fraction of Gorlin syndrome patients get medulloblastoma, which is a tumor of cerebellar origin. This probably relates to the mitogenic role of hedgehog signaling in cerebellar development. The pathway is inappropriately active, cerebellar tumors result, and the blue color here indicates activation of the hedgehog pathway in the tumor. You can see here that the normal foliation of the cerebellum is disrupted, and here the tumor is overgrowing the normal structures. And so basal cell carcinoma, medulloblastoma are both seen at a much higher frequency in basal cell nevus syndrome or Gorlin syndrome. And what I'd like to introduce to you now is cyclopamine, which is the compound shown here, found in this plant, the corn lily, grows in high mountain meadows, about 5,000 feet and up, and which came to the attention of some workers at the USDA in the 50s and 60s when Basque sheep herders in Idaho took their flocks to higher pastures. They experienced outbreaks of cyclopia, like what you see here, and they took their problem to the USDA Poisonous Plant Research Center in nearby Logan, Utah. And at this location, Lynn James, when he was much younger, then camped out in the mountains with a flock of sheep, fed the pregnant ewes various plants at varying stages of gestation, and identified veratrum, the corn lily, as the responsible plant. And this gentleman here then, who's a chemist, worked out that cyclopamine was the compound responsible for this cyclopia that you see here. And as I mentioned earlier, now this is a lamb, a cyclopic lamb. This is a very, very good phenocopy of the sonic hedgehog knockout in the mouse. And so when we analyzed that phenotype, we were able to surmise that there may be a connection between the effects of cyclopamine and the hedgehog pathway, and we guessed that cyclopamine might be a pathway antagonist. That turned out to be the case. I'll just point out on this picture here, James Chen, who's here at Stanford, a faculty member in the Department of Chemical and Systems Biology. When he was a postdoc in my lab, he was involved in helping to demonstrate that cyclopamine indeed inhibits hedgehog pathway activity and does so by acting on smoothened. And one of the first things we learned is that cyclopamine inhibits responsive cells to the hedgehog signal, not production of the signal, which was our first guess actually. And through genetic studies in cell lines, found that cyclopamine acts downstream of patched, upstream of glee, possibly at smoothened. And James then actually made fluorescent and photoactivatable cross-linking derivatives of cyclopamine and used these to show that cyclopamine binds to smoothened. So this is just one example of those kinds of studies, cells that are transfected for expression of smoothened. And you can see that these bright cells here are the cells that were transfected, and they're binding to the fluorescent cyclopamine. This can be competed with a non-fluorescent cyclopamine. With the cross-linking version of cyclopamine, it could also be shown that there's direct interaction between cyclopamine and smoothened. And so cyclopamine acts by binding directly to smoothened and preventing activation of smoothened in this pathway. And just to point out here, while we have this diagram, even if patched is absent, and in this circumstance, of course, pathway would normally be active. This is what happens in the tumors that arise in Gorlin syndrome. Cyclopamine could still act to block pathway by binding to and inhibiting the action of smoothen. And that's exactly the effect that we explored in the context of a mouse model of Gorlin syndrome, which was produced in Matt Scott's lab. And so here is shown the tumor, and here is an allograft of that tumor into a nude mouse, and you can see that it's grown to a pretty good size here, and we were able, by injecting cyclopamine, to cause a regression of these pre-existing tumors, and in some cases to cause complete regression. Now, others have shown that either cyclopamine or other small molecule antagonists of this pathway are also capable of inhibiting the growth of endogenous tumors in this patched heterozygous mutant mice. And this shows that pathway activity is not only important in initiating tumors, but is also continuously required for cancer growth. And of course, this raises the possibility of use of pathway blockade in therapeutic applications. Now, medulloblastoma arises in a number of settings. There are various etiologies. I've mentioned loss of patched in Gorlin syndrome. Another negatively acting pathway component that interacts directly with the Gli proteins is suppressor effused. Loss of suppressor effused also associated with medulloblastoma. There can be sporadic activation of smoothen through specific missense mutations, and this is seen in, all of these things are seen in both mouse and human. In addition, in some other genetic backgrounds, this is a PARP mutant mouse, also lacking P53, which enhances the incidence of medulloblastoma, not only in the PARP mouse, but also in the patched heterozygous mouse, much higher frequency. This also is associated, even though these mutations really have nothing directly to do with hedgehog pathway activity, this also results in high pathway activity in the tumors. This is another genetic background that produces high pathway activity. So all roads, or many roads at least, appear to lead to hedgehog pathway activation in medulloblastoma. And so that makes this this strategy of blocking hedgehog pathway activity a particularly attractive one for this really nasty childhood disease. And we were able to show in collaboration with Jim Olson at the Hutch a number of years ago now that resected human medulloblastomas, primary cells put into culture, are killed by cyclopamine. And this was true in 7 of 7 cases, whereas timatidine, a related compound with no potency in pathway inhibition, doesn't kill the cells, and other types of brain tumors are not affected by cyclopamine, indicating specificity, a specific effect, and not simply general toxicity. Okay, but in this study, we also, and many other investigators also found that only a fraction of medulloblastoma is associated with patch mutation or with mutations at smoothened. And so again, this makes the point that most, perhaps all, cases of human medulloblastoma connect in some way to hedgehog pathway activation. It also raises the question, though, of how pathway is activated in these other circumstances. So in contrast to loss of patched in Gorlin syndrome tumors, there's actually a whole group of tumors in which pathway activity appears to be activated in the same way that it is normally, that is, through presence of the ligand. And perhaps it's the case in some of these tumors that limiting components in the pathway become expressed, and that's something that a number of people are exploring. But in any case, this is now ligand-dependent pathway activation. And some of the tumors in which this is seen include cancers that arise from the endodermal epithelium: small cell lung cancer, esophageal, gastric, biliary tract, pancreatic cancer, bladder cancer, metastatic prostate cancer, and I'm going to say a few words about that. A lot of this work came from folks in my lab and colleagues at Hopkins who have pursued these various diseases as a very special interest and a focus for their research. So I'd like to now show you just a little bit of data in the area of prostate cancer. And these are some very special samples collected by Steve Bova at Hopkins in the warm autopsy program. These are men who have agreed to be sampled before they succumb to the disease, and then very shortly after death, samples are obtained. And these are samples at sites of metastases. And so what you can see here, and these are qualitative RT-PCRs, and these are quantitative RT-PCRs, looking at expression of patched, which I told you earlier is also a pathway target. So whenever you have high levels of pathway activity, you also, in general, see high levels of patched expression. And what you can see is that the levels of pathway activity are rather low in normal prostate or in localized prostate cancer, but in contrast are quite high in metastatic prostate cancer. And you'll note here that the axes are quite different. Let me see. Yes, here we go. The axes are quite different, so that the highest of the localized prostate cancers is some tenfold less than the lowest of the metastatic cancers in patched levels. So again, only a minority of localized prostate cancers displayed pathway activity. All of the metastatic cancers did. And this suggested the possibility that hedgehog pathway activity enables metastasis, and also raised the question of whether perhaps pathway activity indicates metastatic potential. And that was an especially interesting possibility because a few of the localized cancers did show low levels of pathway activity. And this also raised the question of whether this could be useful prognostically. We and collaborators here at Stanford and at Hopkins are involved now in trying to determine whether in biopsies or other samples that can be obtained from men with prostate cancer, whether levels of pathway activity are prognostic and might indicate a potential for metastasis. Okay, I'd like to, and I should say, we've done some studies with cell lines to show that there is some effect of hedgehog pathway inhibition on metastasis of prostate cancer in mouse models. I'd like to shift for a minute or two now to pancreatic cancer to tell you about some recent work that was published by Anirban Maitra and his group at Hopkins, showing that pancreatic cancer metastasis is blocked by hedgehog pathway blockade. And so the methodology here was to make orthotopic grafts of human pancreatic cancer cell lines. And these are very recently derived cell lines coming with a very low passage number from human prostate cancers. And this is one cell line and another cell line here. And the kinds of metastases that are seen, so the grafts are made by introducing cells right into the pancreas, and then metastases are observed typically in the spleen, kidney, liver, and other organs. And with two different cell lines, what was seen is that metastasis was blocked by treatment with cyclopamine. So instead of the all or most of the grafts producing metastases to a whole variety of organs, with cyclopamine treatment, very little metastasis is seen with both of these cell lines. And I should mention that not all pancreatic cancer cell lines show evidence of hedgehog pathway activity. There is some heterogeneity in this disease. It's also true that in vitro invasiveness of these cells, invasiveness into matrigel or membranes impregnated with matrigel, gene expression and of these cell lines are all also affected by cyclopamine treatment. So I'd like to turn now to another malignancy, and this is multiple myeloma. And what we know now from work initially of Bill Matsui at Hopkins, and then we've collaborated more recently in looking at hedgehog pathway blockade here. What we know is that multiple myeloma stem cells can be purified from the marrow of patients, and these are CD138 negative cells. CD138 is a marker for mature plasma cells, and these can be purified, and these cells show clonogenic growth in soft agar, and they can initiate the disease in non-skid mice when they're transplanted. And so that's what's shown here. And these transplanted cells produce the lambda light chain antibody in the serum of mice, characteristic or indicative of the source of these cells from the patient, human lambda light chain, I should say. And what we know also is that these stem cells are dependent on hedgehog activity, because we find that cyclopamine inhibits clonogenic growth and induces differentiation. So this is a clonogenic growth assay, and what you can see is that cyclopamine treatment, or treatment with an antibody that antagonizes hedgehog binding and activity, reduces the number of clones recovered. And furthermore, treatment with cyclopamine causes differentiation of these cells, and that's indicated here by an increase in level of expression of CD138, and also in this side population here, dye exclusion is the property that's exploited here. The number of progenitor cells is dramatically decreased by treatment with cyclopamine. Okay? So, in summary, hedgehog pathway activity is associated with tumor initiation and metastasis in a number of settings. Something that was found in the multiple myeloma situation is that hedgehog pathway activity actually was only present in a subset, a small percentage of the tumor cells. And bulk tumor doesn't always show an immediate response. And this would be consistent with a rather specific effect of pathway antagonism on the cancer stem cells. And I know that you've heard about cancer stem cells and have discussed cancer stem cells at length earlier in this course. And so it may be the case that response to hedgehog pathway inhibition in many settings is best measured by looking at clonogenicity, and that's something that is important to keep in mind in some of these studies. Now, cyclopamine derivatives and other small molecule antagonists are of great interest in industry, and they're under development for cancer therapy at several biotechs and a number of large pharmaceutical companies. What I'd like to tell you a little bit about now in the last few minutes is another approach to therapy to try to streamline the path to therapy in patients, and that is to look for drugs that are already approved for use in humans, to look for drugs that also inhibit the hedgehog pathway. And one reason that this seems possible is that the hedgehog pathway is pharmacologically very accessible. Smoothen is a major target in the pathway. In screens of synthetic small molecules that we have done many years ago now, we found a reasonably large number, more than a dozen structurally unrelated families of small molecules that antagonize pathway and do so by binding to smoothen. They compete with cyclopamine for binding, actually. So smoothen is really a very good target. I think this relates perhaps to the way that it's regulated physiologically. I mentioned that patched looks like a transporter. It may inhibit smoothen by transporting a small molecule that then modulates smoothen. I'm not going to have time to discuss that model and the reasons for at least attempting to test it, although it's of great interest to my lab, I think the mechanisms in this pathway are very interesting. But I think that may be the reason why this pathway and smooth in particular are very accessible pharmacologically. So the way to approach this then was to screen compounds that have progressed at least through phase 1 testing, so there's some information on toxicity in humans. And this was a library that we screened about 3 or 4 years ago. It was assembled by Jun Liu and Curtis Chong in the Department of Pharmacology at Hopkins. And we found many of these. One very striking class of these was microtubule drugs. So all microtubule antagonists are pathway inhibitors. And on the other hand, Taxol, which is a microtubule stabilizer, is not an antagonist. This has led to a project in my lab to try to understand the role of microtubules in signaling. But in any case, these drugs are cytotoxic, and they're not really suitable for long-term treatment. We also found several antihistamines and antidepressants and some antibiotics, but serum levels of these things, the achievable serum levels, might be too low to make them useful. I'd like to tell you just a little bit about itraconazole, which is an antifungal azole in the library, and this had potent pathway inhibition. This is the structure of itraconazole shown here. It's closely related to a number of other azole antifungals, including ketoconazole, which is shown here. You can see that they're very closely related, differing in one part of the molecule here on the right. And yet they have actually very different effects on hedgehog pathway activity, with itraconazole showing a potency that's not too dissimilar from cyclopamine, and these other azole antifungals basically not doing much at these levels. And these are levels, by the way, where they do affect the endogenous target. And so itraconazole is a drug that we're very interested in studying further to test to whether it may be a possible therapy for cancers that involve hedgehog pathway activation. Well, another fact that I'd like to just bring up, or observation, is that there is a very strong and striking correlation between statin use and a decreased incidence of metastatic prostate cancer. Now, this is a somewhat controversial topic in general in the field of cancer. There have been a number of meta-analyses published in journals like Lancet or the New England Journal, which say that statins really don't correlate at all. Use of statins don't correlate at all with incidence of cancer. But if you really look at the data, and if you look at particular cancer types, and prostate cancer is a very striking example, there is a dramatic correlation between use of statins and a reduced incidence of metastatic prostate cancer. And this is metastatic prostate cancer in particular, not prostate cancer in general, but the more aggressive forms of prostate cancer. And this correlation is dependent on the dose used. The higher, the stronger the correlation. On the duration of use, the longer, the stronger the correlation. And the best study here is the one by Platz et al., in which there's a prospective study with a huge number of person years of follow-up, several hundred thousand, and this is a prospective study. And the reason I'm bringing this up is because we had studied some years before this correlation emerged, we had studied the role of cholesterol in hedgehog pathway activity. And I'll show you some of that evidence in a minute, but this did raise the question for us whether this correlation may be because of an effect of statins on hedgehog pathway activity. And so these are some of the data, and what we found was that in cultured cells, if we use cyclodextrin to deplete cholesterol from membranes, this is a trick that cell biologists routinely use to pull cholesterol out of plasma membranes, disrupt lipid rafts, microdomains. If we do that, if we use cyclodextrin and apply it chronically throughout the experiment, with increasing concentrations of cyclodextrin, we see inhibition of hedgehog pathway response. And in fact, if we treat chick embryos with cyclodextrin, and if the chick embryos will make an appearance, they don't seem to wish to do so. Oh well. I'll just tell you that we're able to see holoprosencephaly in these chick embryos. Midline structures are lost, and see some of the same kinds of features that I pointed out and described at the beginning in cyclopamine-treated chick embryos. Well, what about statins? As a matter of fact, if we treat cells with compactin, which is a statin, a commonly used statin, and what we have to do to make this observation work is to pre-treat with some cyclodextrin. And that's because these cells come replete with cholesterol because of prior culture conditions. So maybe we could just focus on this column here and the one and one right next to it all the way to the right. And what you can see is that if we treat both with cyclodextrin, the response is perfectly fine here. But if we add statins here, the response is completely wiped out. And so response to Hedgehog signaling under these conditions is dependent on cholesterol synthesis, which is blocked by statins. And so I'll just leave you with that as a final sort of observation. I think that it would be very interesting, and I'm not sure whether this is happening now, but it would be very interesting to carry out drug trials with statins in prostate cancer. So I guess that's it for now. I would like to, I'm listing here people from my lab and direct collaborators. I've also mentioned some other work which I tried to credit as I went through. James Kim, together with Royu Gong, were involved in the screen of pharmaceutical compounds. David Berman, a lot of the tumor studies. James Chen and UC Taipale in studies of cyclopamine interaction. As I mentioned, James is on the faculty here. UC is back in Finland, where he has a faculty position. David Berman is at Hopkins. He's a pathologist on the faculty in the pathology department there. The studies of multiple myeloma were pioneered by Bill Matsui together with Neil Watkins and a postdoc working jointly in their labs, Craig Peacock. The library I mentioned was produced or assembled by Curtis Chong and Jun Lu's lab. So thanks for your attention.
The preceding program is copyrighted by Stanford University.